Namaste students. In today's session we are going to see about the plant anatomy. In that we are going to see dicot and monocot stem. By this session you would come to know the primary structure of monocot and dicot stem, the tissue differentiation of monocot and dicot, and then the vascular bundles in monocot and dicot, and what are the difference between monocot and dicot stem. By this session you will all know these things. So before entering into the class, we would learn about how to make a session. So whenever we are taking a session, whether it may be a root, stem or leaf, take that stem in the left hand. It should be placed in between the index finger and thumb finger. So once you place it, then you have to make a thin sections using a sharp knife or a blade. So after taking a session, please transfer the session into a water. So after some times select a complete section. So without any damage, the internal sections in which section it is clear, take that section and you have to strain using saffronin. So after straining, add a drop of glycerin and place a cover slip over it and wipe the excess strains and glycerin in that and observe under the microscope. So once you observe under the microscope, the cells inside the stem will be stained with saffronin and you can able to see the structures in clear. So now we shall go to the topic. So dicot and monocot stem. By this diagram itself you can understand the difference between the dicot and monocot stem. There are lot of difference between the dicot and monocot stem here. So we shall learn one by one in detail. The first one is epidermis. So epidermis is the outermost layer of a stem. So it is made up of single layer of parenchymatous. Since it is a shoot system, it has cuticle present in it. Cuticle is nothing but a waxy coat which prevents loss of excess water, and then it is protective in functions. The difference between the epidermis of monocot and dicot is the multicellular hairs is absent in monocot whereas present in dicot, and then the stroma is more in numbers in dicot and it is less in monocot. These are the difference between the monocot and dicot. The next region is the cortex. So the cortex is nothing but the middle region. So this cortex region has a vast difference between dicot and monocot. So in dicot stem, the cortex is divided into three regions. They are hypodermis, middle cortex and inner cortex. So hypodermis is nothing but it is a collenchyma cells. So 3 to 6 layers of collenchyma cells arranged in a arranged and it gives a mechanical support. Next one is the middle cortex. So middle cortex is made up of chlorenchyma cells. So these chlorenchyma cells helps in photosynthesis. So due to the presence of chloroplast. So you all know that chloroplast helps in photosynthesis. So these chlorenchyma cells has chloroplast which helps in the photosynthesis. The next one is inner cortex. So inner cortex is the innermost region of the cortex region. It is made up of parenchyma cells and it helps in the gaseous exchange and food materials. So now we shall go to the monocot stem. In monocot stem, the cortex is not differentiated as in dicot. So it has only the hypodermis region. So hypodermis is compactly packed and multi-layered sclerenchymatous cells. So it is alternatively present. Collenchyma cells are also present. So this chlorenchyma helps in photosynthesis. So this is arranged in a manner like sclerenchyma and chlorenchyma. They are present in between each others. So in monocot only hypodermis is present, all the other regions are absent. So this hypodermis gives the mechanical support here. And then the vascular bundles are embedded in the hypodermis region. Next one is the ground tissue. So this is also seen in the monocot stem. It is not differentiated as in dicot. So in monocot the ground tissue is not differentiated and it is made up of a parenchymatous cells and it is loosely packed with intercellular space for gaseous exchange. Now we shall go to the innermost region. So the next one is endodermis. So this endodermis is peculiar only to the dicot stem and it is absent in the monocot stem. So this endodermis is the innermost layer of the cortex and it is made up of barrel shaped cells and it is compactly packed. This endodermis is made up of starch grains, so it is also called as starch sheet and it is absent in monocot. The next one is vascular region. So the innermost region of both monocot and dicot. In dicot, the innermost region, that is below the endodermis, it has many layers. So all these layers combinedly called as style. So this style consists of pericycle, vascular bundle, medullary rays and pith. So now we shall learn in one by one. So the pericycle. So pericycle is the outermost layer of the style and it is present immediately below the endodermis. The next is the vascular bundle. So the vascular bundle vary from each monocot and dicot. So the vascular bundle of dicot is conjoined, open, xylem is endarched and it is collateral. Now we can go to the xylem. So the function of xylem is to conduct water and minerals and the xylem is composed of tracheids, vessels, xylem vessels, fibers and parenchyma. So here the xylem is differentiated into protoxylem and metaxylem. Protoxylem is nothing but small developing xylem and metaxylem is completely developed xylem. The next one is phloem. Phloem conduct food material. Phloem is also composed of sheath tubes, companion cells, parenchyma cells and fiber. Here also the phloem is differentiated into protoxylem and metaxylem. The arrangement is protoxylem is towards the periphery and metaxylem is towards the center. So this type of arrangement is called as endarch. The next is medullary ray. So in between the vascular bundles there is a ray present in between all the vascular bundles. So that is called the medullary ray. So this medullary ray is also called as pith ray. So it is composed of parenchymatous cells and it is the cells are radially elongated. The next is the innermost region. So innermost region is called the pith. It is made up of parenchymatous cells and the major function of pith is to store the food material. Now we shall move to the monocot. In monocot, the vascular bundle are numerous. As I said, they are numerous and they are scattered here and there. There is no specific arrangement as in dicot. So they are scattered here and there, and then the size also, the size of the vascular bundle also vary. That is, the small vascular bundles are seen in the outer region, whereas the larger vascular bundle are seen in the inner region. Here the vascular bundle is conjoined, collateral and closed. The next one is xylem and phloem. Here xylem consists of protoxylem and metaxylem and it is endarched. Here the xylem is arranged in V-shaped. So I can assume V means 3. So the xylem is arranged in a V shape. Next is the phloem. The phloem is present in the present to the outer of the xylem. So phloem composed of sheath tubes and companion cells. Here the phloem parenchyma is absent. Each vascular bundle is surrounded by a bundle sheet. So these bundle sheet are made up of sclerenchymatose. And in some cases these bundle sheet are absent. Example, in asparagus this bundle sheet is absent. So in you can see the space in between the xylem. So that is nothing but a lacunae, that is a space. So it is seen in monocot stem, that the space is called as lacunae. Now we shall see the difference between the monocot and dicot stem. So here their tissues are given and how it is in dicot and monocot. The first one is hypodermis. So the hypodermis in dicot is collenchymatous, whereas in monocot it is sclerenchymatous. The next is ground tissue. Ground tissue is differentiated here, there it is undifferentiated. Next, the vascular bundle is less in number, uniform in size, arranged in ring, open, bundle sheet is absent. Whereas in monocot, vascular bundle are numerous and the size vary and it is scattered, closed and bundle sheet are present. Secondary growth is present, here mostly absent. So the secondary growth is present in dicot stem and absent in monocots. Pith is present In monocot, the pith is absent. Medullary ray is present in dicot and it is absent in monocot. And you will have a small video clipping to recall this structure of monocot and dicot.
TS of dicot stem. We can observe the TS of a dicot stem through the microscope. The dicot stem has an outermost layer made up of parenchyma cells known as epidermis. The epidermis is covered with cuticle towards the outside. Below the epidermis lies the hypodermis, and it is made up of a few layers of collenchyma cells. Several layers of parenchyma cells that lie below the hypodermis is known as cortex. Endodermis is a single layer of parenchymatous cells that separates vascular tissue from the cortex. The vascular bundles are wedge-shaped and arranged in a ring. Each vascular bundle is conjoined. That is, xylem and phloem occurs in the same bundle. Collateral, that is, phloem lies towards the outer side and xylem occurs towards the inner side. And open, that is, cambium is present between phloem and xylem. Medullary rays are made up of parenchymatous cells between the vascular bundles. Features of identification. Presence of multicellular hair on the epidermis. Presence of collenchymatous hypodermis. Vascular bundles are wedge-shaped and are arranged in a ring. Each vascular bundle is conjoined, collateral and open. TS of monocot stem. We can observe the TS of a monocot stem through the microscope. The monocot stem has an outermost layer made up of parenchyma cells known as epidermis. Below the epidermis lies the hypodermis and is made up of a few layers of sclerenchymatous cells. Ground tissue is composed of parenchymatous tissue and extends from hypodermis to the center. Endodermis is a single layer of parenchymatous cells that separates vascular tissue from cortex. The vascular bundles are numerous and scattered in the ground tissue. Each VB is oval in shape and possesses a sclerenchymatous sheath. Each vascular bundle is conjoined, collateral and closed. Features of identification. Absence of short hair. Presence of sclerenchymatous hypodermis. Ground tissue is undifferentiated. Vascular bundles are oval in shape, numerous and scattered in the ground tissue. Each VB is conjoined, collateral and closed. Xylem vessels are arranged in a V-shaped manner. TS of
So now you could understand the structure of monocot and dicot stem and then the similarities and difference between the cells. Thank you.